Pharmaceutical research generates an immense volume of unstructured information. Every day, disparate lab reports, physician notes, and telemetry streams are crashing into databases. To manage this, the U.S. Food and Drug Administration and the PMDA strictly mandate a standardized technical format for all trial submissions. When sponsors submit customized datasets and document-heavy reports, automated review algorithms fail. A machine cannot parse a PDF narrative to determine if a specific patient's blood pressure spiked on day 4 of a trial. The core engineering challenge is mapping the infinite biological and operational variability of thousands of human and animal test subjects into a single predictable schema. Without a universal data ontology to organize these inputs, a drug's efficacy cannot be statistically proven, rendering regulatory approval impossible. To solve this, the Clinical Data Interchange Standards Consortium created the Study Data Tabulation Model, SDTM. Rather than a simple collection of spreadsheets, SDTM functions as a highly governed relational architecture that forces raw, unstructured observations into a strict tabular framework. This architecture enables automated validation checks, large-scale data warehousing, and direct pooling across independent studies. To understand how the entire framework operates, we have to look at its base component, the atomic unit of clinical data. Mastering SDTM requires understanding this fundamental structural logic, rather than simply memorizing domain acronyms. In clinical research, an observation is a discrete piece of collected information. In SDTM, each observation forms a single row in a database. Every column in that row must be classified into one of 4 strict variable roles: identifiers, the topic, qualifiers, and timing variables. Let's map a clinical example. We start with the identifier. The unique subject identifier column, or USUBJID, receives the value 111. Next is the topic, the central focus of the observation. If we measured body temperature, the vital signs test code column takes the value temp. Qualifiers give the topic meaning. The result column records 37, and the unit column records degrees Celsius. Finally, the timing variable records exactly when the assessment occurred, placing the date 01 Jan 2020 into the date time of collection column. These variable names, like USUBJ, look cryptic because they inherit rigid technical constraints from SAS version 5, which enforces an 8-character maximum for column names in transport files. By forcing every data point through this exact atomic grammar, the system guarantees that highly complex clinical events become perfectly machine-readable. Once an observation is structured, it must be stored. Almost all clinical data is funneled into 3 massive structural silos known as general observation classes. The first class is interventions. This silo captures any treatment or exposure administered to the subject, such as the study drug. The second is events. This captures planned protocol milestones, pre-existing medical history, or unplanned occurrences, like adverse events. The third is findings, which stores the results of specific planned evaluations, laboratory tests, vital signs, or ECG readings. To maintain order within these silos, SDTM enforces a 2-character domain prefix rule. The general term for an event becomes AE term inside the adverse events domain and MH term inside medical history. Each column is further governed by CDISC core variable rules, classifying data as required, expected, or permissible to mathematically control how missing values are handled during regulatory submission. Respecting these 3 classes prevents systemic data corruption, ensuring that physical treatments, biological reactions, and laboratory results are never mixed. But clinical trials are unpredictable. If a sponsor collects data that exceeds the standard columns defined by STDM, an architectural tension arises. You cannot simply insert a new column into a standard domain. The system will explicitly reject it as a structural error. Instead, this non-standard data is routed into supplemental qualifiers, or subqual. This acts as a strictly governed repository for custom variables. To maintain the relationship, the subqual table links the custom data back to the parent record using the unique subject identifier and an internal sequence number. When dependencies exist between 2 entirely different domains, STDM uses the related records, or REL-REC dataset. If a patient is given a specific concomitant medication to treat an adverse event, RelRec draws a direct relational link between the row in the CM domain and the row in the AE domain. These relational fallbacks preserve the rigidity of the core schema, while ensuring that zero clinical data is lost to formatting constraints. As an absolute last resort, sponsors can create custom domains, identified by an X, Y, or Z prefix. However, these carry strict prohibitions. A custom domain cannot be created simply to separate data chronologically, nor can it duplicate the function of an existing standard domain. This fundamental structural logic extends seamlessly beyond traditional pharmaceutical trials, encompassing both non-clinical animal studies and complex medical devices. Medical devices require specific domains to capture static and operational traits, primarily device identifiers, DI, and device properties, DO. The manufacturer, model, and serial number on the unit map into the DI domain under the sponsor device identifier variable. Because it relies on defined observation classes and relational keys, the SDTM grammar is universally scalable. It proves as effective for a mechanical pacemaker as it is for a biological blood draw. When all domains are assembled, the architecture relies on one critical special-purpose domain to hold it together, demographics, or DM. Demographics is the central parent domain. Without a DM record, a subject does not exist. The interventions, events, and findings classes orbit this core, tethering rows back to DM. Surrounding this, trial design datasets lock the subject data into a chronological sequence. This interlocking node map provides a machine-readable reconstruction of the entire trial, ensuring every physical intervention or biological result has a traceable, verifiable digital coordinate. Before any of this data reaches a regulatory agency, the entire architecture must pass rigorous automated compliance checks using software validators like Pinnacle 21. These tools scan the datasets for structural errors, missing core variables, or terminology mismatches. If a data point fails a check due to an unavoidable protocol deviation, it triggers a technical rejection. Valid deviations from the standard cannot simply be ignored. They must be explicitly documented and justified in the Study Data Reviewer's Guide. The strictness of this standard provides a massive operational payoff. Reviewers at the FDA can run standard query code and analysis algorithms across aggregated studies, even if those studies were conducted by competing pharmaceutical companies. SDTM converts thousands of scattered biological observations into a universal language, allowing regulators to verify the safety and efficacy of a drug with mathematical precision and speed.